Hello and welcome to some online class. Good evening friends. Today we shall discuss about spontaneous mutation. Most of the mutations occur spontaneously and they have no known cause. Spontaneous mutation arise mainly due to DNA replication error and base modification. DNA synthesis is a cooperative venture in which many cellular components participate. Therefore, occasionally errors do occur during the replication and distribution of genetic material. Replication error can arise due to mispairing. The fact is that both purines and pyrimidines exist in various structural isomeric forms increases the chance of mispairing during the process of DNA replication. Mispairing during the process of DNA replication predominantly lead to point mutation. So the nitrogenous bases, both purine and pyrimidines, they exist in various structural isomeric forms known as tautomers. So tautomers are the structural isomers. The tautomers are interconvertible. Tautomerization is the interconversion between two specific tautomers, and the phenomenon is known as tautomerism. So tautomerism increases the chance of mispairing during the process of DNA replication. Any type of mispairing between nitrogenous bases lead to point mutation. So both purines and pyrimidines can exist in the form of various tautomers. The tautomerism arises due to the relocation of hydrogen atoms within the compounds. Biologically important tautomers are the keto form and enol form of thymine and guanine. Two nitrogenous base thymine and guanine can exist in keto form as well as in enol form, and keto form is the normal form. Similarly, other two nitrogenous base, cytosine and adenine, they can exist in amino form and imino form, and amino form is the normal form. So due to this tautomerism, base pairing occur between non-complementary bases, and the base pairing between non-complementary bases lead to mutation. Now we shall see how tautomerism contribute to the mispairing between non-complementary bases. Friends, here you can see two types of base pairing arrangement. One is standard base pairing arrangement and the other is anomalous base pairing arrangement. Standard base pairing arrangement is normal, whereas anomalous base pairing arrangement is abnormal, and this anomalous base pairing gives rise to point mutation. What is standard base pairing? Standard base pairing is the normal base pairing that occur between amino form and keto form of complementary nitrogenous bases. Here, this is the amino form of cytosine. Amino form of cytosine is the normal form. Similarly, keto form of guanine is the normal form. So between amino form of cytosine and keto form of guanine, there is standard base pairing. Similarly, thymine keto form and adenine amino form can also base pair normally. So this is another standard base pairing. So normal base pairing occur between keto form and amino form of complementary nitrogenous bases. But anomalous base pairing occur between enol form and imino form of nitrogenous bases. Here there are two examples. This is the anal form of thymine. This is not the common form. And this is the keto form of guanine. Here, the keto form of guanine base paired with thymine, which is not normal. So, the anal form of thymine base paired with guanine, keto form. Similarly, the imino form of cytosine base paired with amino form of adenine. This is another anomalous base pairing. So, whenever anomalous base pairing occurs during the process of DNA replication, that gives rise to point mutation. Friend, in this diagram, you can see how tautomerism causes transition mutation. Transition mutation arises due to tautomerism. For example, these are the two DNA strands. So, replication is taking place, and during the process of replication, these two strands has been separated. And in this strand, this is the nitrogenous base adenine. Adenine has undergone tautomeric shift. So, due to the tautomeric shift, adenine has been converted from amino form to imino form. Later on, we will see that these two strands act as template for the synthesis of complementary strand. So, this template strand is copied in normal manner. So, there is no mutation because the replication has been taking place normally and there is standard base pairing. But this template strand has been copied or has been replicated in a wrong way. How? Now, this adenine has been undergone a tautomeric shift from amino form to imino form. And the imino form of adenine base pair with cytosine. So, base pairing between adenine and cytosine is anomalous base pairing. It is not a standard base pairing. So, here the imino form of adenine has base pair with cytosine, which is anomalous. After this, you see there are two DNA molecules. This DNA molecule is normal. There is no mutation. Whereas this DNA molecule exhibit abnormal base pairing because adenine has base pair with cytosine, which is abnormal. So, during the process of replication, these two strands get separated and this strand has been replicated and that strand has been replicated. So, after the separation of these two strands, the imino form of adenine is converted back into amino form, and amino form of adenine is the normal form. So, after converting back to the amino form, this strand will be replicated in a normal manner by following the principle of standard base pairing. So, this DNA molecule will be normal, there will be no mutation. When this strand will be replicated, you see adenine will base pair with thymine, cytosine will base pair with guanine, guanine will be base pair with cytosine, and cytosine will base pair with guanine. So, this is the newly synthesized strand. And this is the template strand. So, particularly in this DNA molecule, you see previously there was adenine thymine base pair, adenine thymine base pair. But due to this tautomerism, AT base pair has been converted into GC base pair. So, such type of base pairing gives rise to transition mutation. In addition to mispairing and point mutation, small insertion and deletion can also occur during DNA replication. Insertion and deletion occur when one of the DNA strand of the template loops out and is displaced during replication and when the DNA polymerase slips and stutters. So friends, now we shall see that this is a DNA template. So this DNA template will be replicated. Before replication, both of these strands has been localized, separated, and this black color strand has been replicated normally without any deletion or without any insertion. But this red color strand has been looped here. So this particularly looped region will be missed when this particular template is replicated. So when this looped strand is replicated, during replication this looped region or this loop will not be replicated. So automatically there will be small deletion. Insertion can also occur when the DNA polymerase repeatedly introduce nucleotides which are not present on the template strand. So insertion as well as deletion during DNA replication can cause frame shift mutation and also amino acid insertion and deletion in the polypeptide chain. In addition to this, replication slippage can also give rise to spontaneous mutation. Replication slippage occur anywhere in the DNA molecule, but it is more common in the regions containing repeat sequences. Such type of repeat sequences are the hotspots for mutation, and in some cases it contributes to some of the hereditary disease like Fragile X syndrome and Huntington's disease. Replication slippage involves misalignment of DNA strands that commonly occur during the replication of repeated sequences. Two of the most common causes of spontaneous mutation are depurination and deamination. Depurination is the loss of a nitrogenous base from an intact DNA double helix, and most frequently the base is a purine. It is either adenine or guanine. The nitrogenous base is lost when the glycosidic bond that link the nitrogenous base with pentose sugar is broken. So the breakdown of the glycosidic bond leaves a apurinic site in the DNA double helix. And when this apurinic site is not repaired, then during the process of DNA replication, the enzyme DNA polymerase can incorporate a nucleotide at random that increases the chance of spontaneous mutation. What is deamination? Deamination is the loss of amino group, and the process of deamination, it can be spontaneous or it can be induced as a result of treatment with certain mutagens like nitrous acid. By the process of deamination, the amino group of cytosine and adenine is converted into keto group. So this conversion of amino group to keto group causes the conversion of cytosine to uracil and adenine to hypoxanthine. And you know that uracil base pair with adenine and hypoxanthine base pair with cytosine. When there is deamination and cytosine is converted into uracil, in that case there will be a transition mutation, and the transition mutation involves the transition of GC base pair to AU base pair, and in the next round of DNA replication, AU base pair will be converted into AT base pair. Similarly, hypoxanthine is another nitrogenous base which is formed due to deamination of adenine, and hypoxanthine base pair with cytosine. So whenever such type of deamination occur, in that case AT base pair can be converted into GC base pair. So the process of deamination contributes to the chances of point mutation. In addition to deep urination and deamination, there are certain other reasons that causes spontaneous mutation. One is oxidative damage, other is transposon. Damage to the DNA can be caused by reactive oxidants. These reactive oxidants are the byproducts of normal cellular process. Examples of reactive oxidants are reactive oxygen species. Various reactive oxygen species that are produced during normal aerobic respiration are superoxide radical, hydroxyl radical and hydrogen peroxide. These reactive oxygen species are a constant threat to the integrity of DNA, and such type of reactive oxidants can produce more than 100 different types of modification in DNA, including modification of nitrogenous bases, loss of nitrogenous bases and single-stranded breaks. So this is the effect of oxidative damage on the DNA. Last is transposons. Transposons are the transposable elements, and the transposons are the genetic elements that move within or between genomes. Transposons can also cause chromosomal damage, including double-stranded breaks, inversions and translocations. Such transposable elements are very natural. They occur in all organisms from bacteria to human beings. These genetic elements move within or between genomes. And transposon can act as naturally occurring mutagens. Mutation occur when a transposon inserts into a protein coding region of a gene, thereby causing a change in the reading frame, and it can also introduce a stop codons. Similarly, when the transposons are inserted into the regulatory region, then they disrupt the normal expression of the gene. So friends, this is about spontaneous mutation. For more updates, please subscribe my YouTube channel, Sam Online Class, and please share your comments. And thank you very much. See you again. Bye bye.